So you have probably heard of muscular dystrophy. But how does that differ from myofibrillar myopathy? Well, the word myo means muscle, and pathy means disease. So technically, all muscle diseases are some form of myopathy. Myofibrils are the long, rod-like organelles found within muscle cells, muscle fibers, that are responsible for muscle contraction. Myofibrils are tightly packed inside muscle fibers and run parallel to the length of the cell. But as it relates to the various forms of muscular dystrophy, there are fundamental differences. Muscular dystrophy (MD) refers to a broad group of inherited dystrophinopathies and related disorders. For example, Duchenne-Becker, limb-girdle, facioscapulohumeral, myotonic, congenital MDs, while myofibrillar myopathy (MFM) refers to various subtypes of congenital myopathies, structural myopathies. Both MD and MFM are muscle-wasting diseases that appear very similar on the outside, but differ greatly at the cellular level. The core genetic difference between the two are genetic mutations, or defects, that regulate protein production in the muscle cells. Muscular dystrophies are caused by mutations that break or remove a protein that anchors or protects the muscle membrane, which causes loss of function. Myofibrillar myopathies are the results of mutations that produce a misfolded or defective protein that clumps inside the muscle fiber, gumming up the internal skeleton, dominant negative or toxic gain-of-function effect. So, in simple terms, MD is caused by a loss of protein production, proteins of the sarcolemma muscle cell membrane or extracellular matrix, whereas MFM is caused by not the loss of protein production, but rather the production of misfolded or defective proteins that clump inside the muscle fiber, also known as protein aggregates, that eventually kill the cell that made them. To understand the process of misfolded proteins and protein aggregates, you must first understand what proteins are and how they are made. Proteins are long, wiggly strings of amino acids linked together. There are 20 different kinds of amino acids. The information in the DNA determine how these amino acids are assembled and folded into proteins. Example, the DMD gene contains the recipe for dystrophin. The DAY gene contains the recipe for desmin. The gene is copied into a temporary messenger molecule called mRNA. Think of mRNA as a photocopy of the recipe that can leave the nucleus. It travels out into the cytoplasm, where the protein factories are ribosomes, tiny protein-making machines, read the mRNA code 3 letters at a time. Each 3-letter codon tells the ribosome to add one specific amino acid. Transfer RNAs tRNAs bring the correct amino acids like delivery trucks. While a muscle cell is dominated by just a few types of structural proteins by weight, it requires a diverse library of roughly 6,000 to 13,000 different proteins to function, repair itself, and produce energy. As the chain of amino acids comes off the ribosome, it starts folding into its correct 3D shape. Chaperone proteins like bicrystalline help it fold properly and prevent tangling. The finished protein is carried to its correct location in the muscle fiber Z-disc, sarcomer membrane, etc., and locked into place with other proteins. Errors in the genetic code can produce misfolded or defective proteins. Most small proteins fold in less than 1 second. Giant muscle proteins can take many minutes and usually need help. Protein folding is the spontaneous but chaperone-assisted process by which a linear amino acid chain collapses, driven largely by the hydrophobic effect, into a unique 3-dimensional shape that allows it to perform its biological job when even one amino acid is wrong in certain muscle proteins. The chain gets trapped in misfolded aggregates instead of reaching its correct structure. So in myofibrillar myopathy, you not only lose the use of a critical protein because it is defective, but those defective proteins clump together and eventually kill the muscle cell that produced it. Thanks for watching, and we will see you next time.